Standard polymerase chain reactions typically require hours to amplify genetic material sufficiently for detection. However, modern rapid protocols achieve results in minutes by compressing thermal cycles and optimizing reagent kinetics. This acceleration relies on precise engineering of temperature transitions and enzyme efficiency rather than altering the fundamental biochemistry. After that first surprise, we can slow down and trace the mechanism behind how can a PCR find a virus in minutes from the first physical step, so the effect stops feeling mysterious and starts feeling engineered. The process begins by identifying unique genetic sequences that distinguish a specific virus from all other biological material. Scientists analyze the viral genome to locate conserved regions, where nucleotide patterns remain stable across different strains and mutations. These conserved gene segments serve as reliable anchors because they are essential for the virus's replication machinery and cannot easily change without compromising viability. For instance, certain structural protein genes often maintain high sequence similarity even when surface proteins evolve rapidly under selective pressure. By designing primers that bind exclusively to these invariant regions, detection systems ensure specificity while minimizing false positives from host DNA or environmental contaminants. This precise targeting establishes the foundation for subsequent amplification steps by confirming which genetic templates require conversion and replication. PAUSE 0.8 Standard polymerase chain reaction relies on DNA templates to function correctly. However, many viruses like influenza or SARS-CoV-2 contain genetic material in the form of RNA rather than DNA. This structural difference prevents standard PCR enzymes from recognizing and amplifying viral RNA directly. To overcome this limitation, we introduce an enzyme called reverse transcriptase into our reaction mixture. Reverse transcriptase acts as a molecular machine that binds specifically to single-stranded RNA templates. Once bound, the enzyme catalyzes the formation of phosphodiester bonds between deoxyribonucleotides. This process synthesizes a complementary DNA strand using the viral RNA as a physical guide for sequence accuracy. Think of this step like converting a handwritten note into typed text so that a standard printer can read it. The resulting molecule is now double-stranded and compatible with conventional PCR amplification protocols. Without this enzymatic conversion, the detection system would fail to identify the viral genetic signature entirely. This preparation phase sets the stage for precise primer binding in our subsequent analysis steps. PAUSE 0.8 The precision of polymerase chain reaction relies entirely on the molecular recognition capabilities of short synthetic DNA fragments, known as primers. These primers are engineered to match a unique sequence within the viral genome, ensuring they bind exclusively to that target region and no other genetic material present in the sample. This binding process is driven by hydrogen bonding between complementary nitrogenous bases, where adenine pairs with thymine and cytosine pairs with guanine through specific thermodynamic interactions. Consider a scenario where a primer sequence matches only one location within a complex mixture of human DNA and viral RNA fragments. The thermal stability of this interaction is governed by the melting temperature, which dictates that binding occurs only when the environment allows sufficient energy for stable hybridization without random association. The selective annealing acts as the critical checkpoint that prevents nonspecific amplification and ensures the reaction targets the correct pathogen sequence with high fidelity. Once these primers have successfully annealed to their specific targets, the reaction must undergo controlled thermal cycling to drive exponential replication. PAUSE 0.8 The core mechanism of polymerase chain reaction relies on a precisely controlled thermal cycling process that drives molecular state transitions through three distinct temperature phases. First, the reaction mixture is heated to approximately 95 degrees Celsius, which provides sufficient kinetic energy to break hydrogen bonds between complementary DNA strands and achieve complete denaturation. Next, the temperature drops rapidly to around 55 degrees Celsius, allowing short synthetic primers to locate their specific target sequences through a process called annealing, where base pairing occurs. Finally, the system stabilizes at 72 degrees Celsius, which represents the optimal activity range for heat-stable DNA polymerase enzymes to synthesize new complementary strands from available nucleotides. This cyclical pattern repeats continuously, with each complete cycle doubling the quantity of target genetic material through predictable reaction kinetics rather than random molecular interactions. The temperature time graph displayed illustrates how these three phases correlate directly with specific molecular behaviors required for successful amplification without any clinical data overlays. Pause 0.8 The core capability of PCR to detect viral material within minutes relies on a specific mathematical property known as exponential amplification. Unlike linear growth, where one unit adds another, this process doubles the target sequence during every single thermal cycle. Starting with just 2 copies of a genetic segment, the first cycle produces 4, followed by 8 in the second round. By the 10th cycle, we have reached over 1,000 copies from that initial trace amount. This compounding effect means that after 30 cycles, a single molecule can theoretically generate more than 1 billion identical sequences. To visualize this progression, consider how a population of bacteria might grow versus a chain reaction, where each link creates 2 new links simultaneously. The resulting growth curve rises slowly, at first before shooting upward in a steep vertical trajectory that defines the detection threshold. This rapid multiplication transforms an undetectable quantity into a measurable signal without requiring additional biological material from the patient. Pause 0.8 The detection of viral genetic material relies on specialized fluorescent probes designed to interact with the target sequence during amplification. Each probe contains a fluorophore molecule attached to a quencher, which absorbs light energy and prevents emission when in close proximity. As the polymerase enzyme extends the DNA strand, it encounters the bound probe and cleaves it through its exonuclease activity. This physical separation moves the fluorophore away from the quencher, allowing the molecule to emit photons upon excitation by the instrument's light source. The intensity of this emitted signal increases proportionally with each cycle as more copies of the target sequence are generated and cleaved. For example, a single viral genome can produce millions of fluorescent molecules within minutes through exponential amplification cycles. This optical output provides a quantitative measure of the reaction progress without requiring any diagnostic interpretation at this stage. Understanding these optical signals requires precise hardware optimization to capture and quantify the emitted photons accurately. Pause 0.8 The speed of a polymerase chain reaction depends fundamentally on how quickly we can move heat through the sample. Traditional thermal cyclers use thick metal blocks that require minutes to change temperature, but rapid instruments employ thin aluminum or silicon substrates with integrated heating elements. This design minimizes thermal mass so that the block reaches target temperatures in seconds rather than minutes. Simultaneously, specialized reagents contain polymerases engineered for stability at high denaturation temperatures and inhibitors that prevent enzyme degradation during rapid cycling. For example, a standard reaction might take 40 cycles over an hour, whereas optimized systems complete 30 cycles in under 10 minutes by reducing ramp times to less than 1 second per degree. These engineering constraints allow the machine to maintain precise thermal control without compromising the integrity of the biological components involved. The result is a system where heat transfer rates and reagent durability work together to compress the timeline of amplification significantly. Pause 0.8 Rapid PCR detection relies on identifying unique genetic sequences, converting RNA to compatible DNA forms, and utilizing optimized thermal cycles. The process achieves speed through engineered hardware improvements and precise reagent kinetics that maintain fidelity while reducing time.